I'll briefly introduce him. You can tell by his accent that he's not from these parts. He studied in Pretoria. He's South African, studied in Pretoria, did a bachelor's and a master's in genetics there. And I guess, I don't know, South Africa is a wonderful place, but maybe the weather was a bit too warm for him. So he moved very far north to Cambridge, where he spent a long time, PhD and a postdoc, working on these kinds of things with Chris Jiggins and friends. And then a few years ago, he moved even further north to Edinburgh, where the weather's meh. But he holds a Royal Society Fellowship there, which is a quite prestigious thing in the UK. Look forward to your talk.
Thank you. Yeah, thanks so much, Yannick, and thank you so much again to the organizers. This is a brilliant meeting to be at. Yeah, so I guess the need for an introduction keeps decreasing as we go on. So I'm going to just very briefly pull up this picture again that we saw yesterday in Emma's talk. And here I want to highlight in particular the fact that supergenes are quite often maintained not by a single structural variant, but by multiple variants. Oops. And so we could call these, that's the star, there we go. We could call these complex supergenes. And one of the interesting features of these is that people have pointed out that this could come about through extending recombination suppression and perhaps accumulating further loci. But something I want to point out is that this also allows maintenance of more than 2 divergent haplotypes or alleles. And that may be of value in certain situations. And the other interesting thing we've seen, at least in these 2 examples here, these are the fire ants that Laurent talked about and Heliconius pneumata that we heard about earlier from Mathieu. The breakpoints are often nearly identical or as close to identical as we can tell of these different inversions. So this reuse of those breakpoints, and that may come about through repetitive DNA in those regions if it's reused in creating these inversions. And one of the ways that can happen is through duplication followed by non-homologous crossing over, leading to that inversion. But something I wanted to point out, or I want to make a point later in this talk, that perhaps there are other ways that duplicated DNA can contribute to recombination suppression. So that brings me to the study system, which is Danaus chrysippus, this African monarch, which is one of the milkweed butterflies. You probably know its American cousin, which is famous for migration. I hope to convince you today that this is the more interesting member of the genus. It is known for this extensive polymorphism. These samples were all collected in a single location in DRC. But this is probably not a balanced polymorphism, but rather a very large hybrid zone. And interest in the genetics of the species goes back to about the 70s, where my collaborator David Smith and Cyril Clarke did crosses, and they found that all of that variation is controlled by 3 loci, called A, B, and C. A controls this white hindwing patch, B controls dark and light, brown and orange background, and C controls the presence of a forewing tip. And they also showed that B and C are tightly linked, but there is some crossing over between them, and David later started calling this a supergene. We've been trying to understand the geography of this species a bit better, and thankfully it's extremely common, so we can use not only museum specimens that David has gone through over many, many decades, but also citizen science data from places like iNaturalist. And so we've been making these maps of variants at these different alleles. And we see very nicely defined locations for certain alleles, such as the C locus here in East Africa. But we also see large regions of polymorphism. So as you move west from East Africa, you find a hybrid zone for the C locus as well as the A locus. And in the region of around Uganda and Rwanda, around here, you can also find some polymorphism at this B locus. And that's probably why you get such extensive polymorphism in DRC, as I showed in that previous slide. But we think probably what's going on here is local adaptation more than preservation of polymorphism. And that shows up very cleanly in these plots of FST. This is FST across the genome between pairs of populations. And one of the obviously very striking things is that FST is nearly 0 across most of the genome. despite the fact that these are sampled thousands of kilometers apart. So this is obviously an extremely dispersive species. And these narrow, or quite broad, actually, peaks of FST suggest local adaptation. And the one I'm going to be focusing on for the rest of this talk is here on chromosome 15. We call this the BC locus because when we do association analysis, we find that it associates with both the B and C loci. And then the last thing to point out on this slide is that there's a peak of FST in 3 pairwise comparisons between East Africa and West, between East and South, and between South and West Africa. So that suggests that there aren't simply 2 variants at this locus segregating. And in fact, we can use a kind of clustering algorithm that I'm not going to go into now, but happy to talk about with anyone, to slide along the genome and paint by ancestry. But we only paint by ancestry in parts of the genome where there's enough signal to differentiate groups. Otherwise, we paint that window gray, indicating that there's not enough signal. And we can pick out homozygotes for 3 divergent alleles at this sort of central region on chromosome 15. And we can also pick out all 3 types of heterozygotes. These tend to be more common, of course, in that hybrid zone region. And this suggests that we don't have any sort of recessive lethals or anything like that. But we don't really know about the relative frequencies of all of these just yet. It also obviously suggests that we have recombination suppression over a large region of this chromosome. Geographically, these things tend to make sense. In East Africa, we have 1 allele, in West Africa, another. In Southern Africa, we have the 3rd, but you can find heterozygotes. And then this selection of heterozygotes here come from around Nairobi, where you start to get toward the more polymorphic zone. One of the surprises is that in North Africa and Southern Europe, you found the same allele as West Africa, despite the fact that you actually find 2 morphs there. And we think that what's going on here is actually a gene conversion event that has brought some variation from this allele into that one, which I'm not going to say too much more about now. We're still really digging into that data and trying to get some more sequence data to look at that. But we do have some very conclusive evidence that crossing over can happen in this supergene, or perhaps crossing over and gene conversion. And that is in the existence of these very mosaic-looking alleles, both in the hybrid zone and outside it. Perhaps most obviously, these 2 alleles that are almost inverse of each other. One goes blue to red to blue, and then the other goes red to blue. These were found in a small island population here in St. Helena. So we don't yet know what these recombinant alleles mean. Are they simply unfit, or is there more to the story than that? Again, that's still work that's ongoing for us. Really, the question that I'm going to talk about for the rest of this talk is what's the structure of this sort of probably a complex supergene going in, since we have 3 different alleles, and what is suppressing recombination. And this work is almost all published in a paper that will come out in that special issue you heard about from Emma yesterday on Monday, with big contributions from both Rishi and Kanwook here. So we wanted to assemble each of these alleles as a chromosome scale assembly, and we aimed to do this trio-based assembly where we use k-mers to pool long reads based on short read data from each of the parents into 2 haploid pools, and that enables you to assemble 2 haploid genomes. So you can use 1 heterozygote and hopefully get out both alleles of a supergene. We later found out that even without the parents, there's so much heterozygosity in these butterflies that actually, using PacBio data, you can pull out haploid assemblies even without using k-mers. So that was a nice lesson to learn. And to sort of skip over the sort of technical details of all of that, we came out with an assembly for each of our 3 chromosome supergene alleles. I hope you don't mind me calling them alleles. We actually have 2 assemblies for 2 of them, so we're very confident in these, and we can use that same kind of chromosome painting logic to make sure we've not assembled a mosaic. And to start to understand the structure, we thought, rather than compare them to one another, because we expect quite complex changes, we'll first compare them to the outgroup. And there's an outgroup species which we think has not undergone structural change, so should represent the ancestral state, and that's the American monarch, Danaus plexippus. And the first obvious thing you'll see is that its chromosome 15 is much shorter. And when we line up bits that are in synteny, this is using Minimap2 to find alignments, and then I wrote quite a simple algorithm that looks to connect alignments into blocks of synteny, which I'm happy to talk about with anyone interested. We can find nice synteny on the two ends of the chromosome, and we can also find inversions. There's a big one there, a second one a bit smaller, and then a third smaller one further along. So 3 inversions in this allele, but we haven't yet accounted for all of this extra DNA in this longer chromosome. And that's all due to this last little bit over here, which is not only inverted once, but it is present in many different duplicated fragments, most of which are inverted and spread along the length of the chromosome. So yeah, a much more complex sort of series of events here than we see in some other cases. And we see this in the other alleles as well, that this region in light green is duplicated multiple times in multiple fragments here with a different combination of fragments to that one. In this case, there is also an inversion, but only of part of that first inversion we see in this one, so sharing one of the breakpoints. And the other parts are not inverted. Those are collinear with the ancestor. And in the third one, again, we see this big expansion of that one region. That's about a megabase region, by the way. So we're increasing the chromosome length here by something like 5 megabases. And in this case, actually no additional inversions. So these are the 3 alleles, and it's not too surprising that they don't really recombine with one another, looking at the number of different structural changes. We also started to suspect that these duplications may have predated the inversions. So that was the next question, is how do we get a handle on the timing of these events? Dating paralogs is extremely challenging because there are processes like concerted evolution that can cause them to converge. So rather than looking at their sequences, we thought we'd try to look at copy number through the phylogeny. There is Illumina data available for not only our morphs and the outgroup, or that ancestor, that distant relative plexippus, but several other Danaus species. And then we sequenced a more deeper outgroup as well, just to make sure we can root this tree correctly. So this is a species tree based on about 5,000 genes made with Astral. And it's very well supported as long as we ignore our very messy chromosomes. So this is made using clean chromosomes. And so then what we did is, for each gene on chromosome 15, assuming that this Danaus plexippus has a single copy of each of those genes, we then could estimate copy number using Illumina read depth data. So what we're seeing here, each black line is the estimated copy number for a gene based on aligned reads, usually from several individuals per species. So that depth that we see here in Danaus plexippus shows that most of the genes are single copy. The ancestor, the outgroup things are a bit messier, but you get poor read mapping back there because it's quite a divergent species. So I think that's mostly just noise. And then in our 3 morphs, we see these big spikes of increased sequence, increased copy number for about 10 genes. And that's within that light green region that we've seen duplicated many times. Adding some species onto this, these other 2 relatives of Danaus plexippus, this American clade over here, all have single copy in that region. Whereas these 3 species here, which are Asian, African, and 1 American species, all have increased copy numbers in that region. Not the same level of increase in copy number as we see in Danaus chrysippus, but still higher than the background. So this suggests that at least the initiation of copy number change must go back to at least this point in the phylogeny. And we can date that point using synonymous sequence divergence and a calibration point on the deeper tree to about 7.5 million years ago. So quite old duplications in this tree, and we don't know about color, much color polymorphism in these other species, so we don't really know what drove these. And so there's a number of questions that kind of spring into my head at this point. What are those 10 genes? What drove the duplications? How variable is copy number within populations? Is selection acting on copy number, or are these duplicated redundant copies degenerating? Most of these questions we haven't yet gotten to. I can tell you that there's nothing very obvious in those 10 genes. There are some with immunoglobulin repeat domains, but not sure what that should mean in arthropods. I can say a bit about degeneration. We have looked at transposable element content in these chromosomes. And we did this by just sliding along the genome and looking at the number of bases falling into different repeat classes. And that's what this looks like. And indeed, we see elevated repeat content in those bits representing the duplicated fragments of that expanded region, and particularly for line elements, shown there in a sort of orangey color. And we see that in each of the 3 divergent alleles, that where we have those duplicated fragments, we tend to see increased line elements. So this could be some kind of duplication, post-duplication accumulation of TEs. But actually, when we look at that Danaus plexippus, in that light green region, we also see an increase in some TE content. And that is a region that we think has never duplicated in that lineage. So it could be that actually the high repeat content may have triggered some of this expansion. And perhaps both things are going on. That repeat content triggered the expansion, and further repeats have accumulated in these redundant copies. So moving on from those repeats, we also wanted to look at the inversions and ask where in the tree they arose. So we have these 3 regions that are inverted in kind of clean single copy inversions, so we can use the sequences to reconstruct trees in that case. Again, we haven't tried to look at sequence divergence, but rather just, as we saw with the fire ants, compare the inversion tree to the species tree and see what that tells us about the age of the inversions. So this is for the first inverted region, which actually has 2 events, one here and then one there. And the most striking thing here, we're looking just at this clade in gray here, is that one of the alleles in Danaus chrysippus is closer in sequence content to 2 other species than it is to the other 2 alleles. So we interpret this as an ancient polymorphism rather than as introgression. And part of the reason for that is that one of these is an American species and one is found only in Australia. And so we think it's most likely that this one of the 3 alleles of this, or one of the inversion alleles, sort of left the old world sort of towards America, going westward, and then also towards Australia a bit later, going eastward, rather than introgressing across those long distances. So we think that these are sort of established as those species established. But yeah, we are still open to the possibility of 2 introgression events. The second inversion over here has very, very similar structure. Again, one of the morphs is closer to those other 2 species. And so we suppose that there was a polymorphism with both of these 2 inversions back here, which we date to about just over 4 million years ago. So again, old inversions, which is not uncommon for supergenes, but much younger than the duplications, which go back to 7.5 million years. So probably the duplications were there first. The third inversion is the youngest of all. It's also discordant with the species tree, but for a different reason, and that's because the southern African morph here, Orientis, is diverged from the 2 others, as well as this Australian species here. So we think this one also dates back to before that speciation event. So yeah, I think it's quite likely that the duplications occurred first, and that triggered some of these inversions. Two of the inversions are surrounded on both sides by copies of these duplications. And then one of them is, on one side, has these sort of green duplicated fragments. So it seems quite likely that there's non-homologous pairing going on, causing these inversions. So a final piece of evidence about the age of these duplications comes when we line up the alleles to one another. So we can find the collinear bits, as you'd expect, and we can also find these inversions where they exist, as we'd expect. But when we look at the bits corresponding to these duplicated fragments, of course we expect quite a lot of chaos, and we see that. But what we also see is that a lot of these duplicated fragments are not finding a sort of orthologous copy in the other alleles, especially this one at the top here. And we think that is because, so what we've used a sort of quite stringent threshold here of about 10% sequence divergence. And we think that some of these paralogs are just super diverged from one another. And if you have loss of different paralogs in different alleles, then you will no longer have one that you can necessarily pair up with. So just to expand on what I'm saying there, getting close to the end here, we have a model in our minds where we have duplications in the past that then, given enough time, they could have fixed in the population, and then given enough time, can diverge from one another. And this divergence can happen at single bases, but also through things like deletions because of the redundance in some of these genes. And then, if we have independent losses of some of these redundant copies in different individuals, we can have a situation that when we come back together in a heterozygote, individuals may fail to pair up and undergo crossing over, not specifically because of inversions, but just because the sequences have had so much time as paralogs to diverge from one another that there's just insufficient homology. So we think that this may be an additional mechanism for suppressing crossing over, and this may have actually been the first mechanism suppressing crossing over in this species that predates those inversions. So just to conclude, we find these complex rearrangements that can maintain multiple alleles, and in this case, the duplications occurred deep in the past and probably triggered the subsequent recombination suppression. We have lots more unanswered questions here. These include things like what drove those duplications, how common is this mechanism that we've proposed, is this the only case or will there be others? And then, of course, the thing that I'm really interested in getting into, what is going on with those recombinant haplotypes we find in the wild? Are they selected against, or do they enable occasional exchange of alleles between, or exchange of variants between alleles? And yeah, with that, I'll just say thank you to a large number of people, especially Rishi and Kanwuk, and all of these funders, most importantly the Royal Society, that have been very generous to me. And thank you all.